Hi everybody, for today's project we'll be extracting our own DNA. DNA is the reason why everyone is different. It determines things like your hair color, your eye color, and height, along with many other things. This DNA can be found in almost every cell of your body, and today we're going to be taking it from our saliva. The engineering that we're going over today is going to be biomedical engineering. And so biomedical engineering focuses on creating devices and machines and medicine that improve people's health. Things like prosthetic arms, legs, and other limbs, machines for surgery, and new medicines.
The cell membrane is a wall that only allows certain things to pass through. This is like a security checkpoint at the airport. The nucleus is a very important part of the cell. The nucleus is the brain of the cell, which makes it the control center. This is also where DNA is stored, which is what we are extracting today.
Two terms that are good to know are hydrophilic and hydrophobic. Hydrophilic simply means water-loving or attracted to water. For example, salt or sugar. Hydrophobic means water-fearing or wanting to get away from water. An example of this is vegetable oil. Enzymes are things that speed up chemical reactions, so they help the cell do its job faster. It's kind of like drinking an energy drink and feeling like you run faster.
So what is DNA and why would we want to extract it? DNA is what makes you you. DNA is specially coded information that tells our bodies what to do every day even when you're sleeping. Scientists love studying DNA because it helps us understand how humans work. DNA can be used to do a lot of things, including identifying people, treating diseases, and even at crime scenes. Imagine a very, very tall ladder. A ladder has two poles on either side and rungs in between. DNA molecules look exactly like a ladder, except it is twisted like a ribbon. Scientists call this twisted ladder a double helix. The backbone of the helix is made up of strong molecules like sugars that help keep DNA sturdy. The rungs inside are actually the information that makes us all unique. These rungs are called bases, and there are 4 bases that can be mixed and matched to make different patterns. The 4 bases are A, G, C, and T.
The materials that are required for this lab are 1 small plastic beaker, 1 pipette, 1 skewer, 1 test tube, as well as 1 plastic bag. Additionally, you will need a light-colored sports drink, some pineapple juice, some cold 90% isopropyl alcohol, and some liquid dish soap or any type of detergent. In case you don't have any pineapple juice, you can substitute it for strawberry, blueberry, or plum juice. If you don't have any juice in general, smash fresh fruits in a plastic bag and run it through a sieve or coffee filter. If you can't find 90% isopropyl alcohol, you can concentrate 70% isopropyl alcohol into 90%. Pour the 70% alcohol into a jar that has a tight lid. Pour some non-iodized salt into the jar until there is a 1:8 ratio of salt to alcohol. Close the lid tightly and proceed to shake the jar. There will be super concentrated alcohol that is about 95% on the top. There will be a clear line between the super concentrated alcohol and the salt water.
Hey guys! So now that y'all have learned about what DNA is and where you can find it, let's go ahead and extract some. So first things first, you're going to need a beaker, or you can use a glass. You're going to need a pipette or a dropper, and if you don't have one, feel free to use a good old spoon. And then you're also going to need a light-colored sports drink. I'm using Gatorade, but you're welcome to use whatever you guys like. And there's also some dish soap, pineapple juice, some cold isopropyl alcohol that's 90%, and a skewer to grab your DNA at the end. So first we are going to take some of our Gatorade and pour about half an inch of Gatorade into our cup. That looks about right. And so what the Gatorade does is it has salt in it, and so it's able to break the cell membrane, which is the covering outside of the cell, and that way we can access the DNA. And so next what you're going to do is you're going to be swishing this in your mouth for 2 full minutes. And then after that, you can just spit it right back out into the cup. I'm gonna go ahead and set up my timer. And go. Okay, 2 minutes is up. And so now what we're gonna do is we're gonna take our handy dandy dish soap and we're gonna add about, we're gonna fill this all the way until it's halfway full. And so what the dish soap does is it separates the cell membrane, which is like the outer covering of your cell, from the DNA, which is what we want to extract. My dish soap seems to be oozing out very slowly, but hopefully yours is faster. Okay, maybe we can go a little more. Okay, that should be good. That looks like half to me. And what you're gonna do now is you're gonna gently shake it side to side like that. You wanna make sure you're not creating any extra bubbles. Okay, that's what it looks like right now. You can see that there's 2 distinct layers. And so now what we're gonna do is we're gonna take this pineapple juice. Okay, so we're gonna add 10 drops of pineapple juice. Two, 3, 4, 5, 6, 7, 8, 9, last one. Perfect. And so after we've added that, we're also going to do the same thing. We are going to gently shake this side to side. Okay, and now we're going to take our isopropyl alcohol and I'm going to be adding 10 drops of that as well. One, two, three. Four, 5, 6, 7, 8, 9, and 10. Nice. Okay. And now we're going to shake that. And now the easy part, we're just gonna let it sit there and wait for a minute. Okay, so it's been a minute, and ideally the alcohol should be making the DNA into this little white clump so that we can see it. And right now, this is what it looks like. There's a little tiny white thing there, but I don't know if I can pick it up, but we'll go ahead and try it out anyways. So you're gonna take your skewer and gently put it in and turn slowly in one direction. And if you're lucky, you can pick up your DNA, but if not, like mine's didn't work, it's totally fine. It's just because you might not be using the exact measurements, or your isopropyl alcohol isn't cold enough. And you know, in my case, I was using a spoon to do the measurements, so there's definitely some inaccuracy there. So it's okay if it doesn't work out. Just take it as a learning opportunity. And I hope you guys have a lot of fun with this experiment. It's really cool. And yeah, see you guys later.
Here are some helpful tips to make sure that you get the best results when extracting your DNA. First, make sure that you use cold isopropyl alcohol. Make sure that the measurements are as exact as possible, and keep the materials in the order that you need them to avoid making a mess.